guys. So let's go ahead and start on our tricky topics for chapter 12. So as a reminder, the videos I will be covering are Meselson and Stahl, leading and lagging strands, bacterial DNA replication, specifically looking just at that elongation step, and then looking at the eukaryotic DNA polymerases. This video is only over the Meselson and Stahl, so if you want help with any of those other things, feel free to watch those videos. So let's go ahead and get started. So we're going to be looking at Meselson and Stahl's experiment. And before we start that, we got to talk about why they even did their experiment. So Meselson and Stahl really wanted to know how DNA replication was occurring. And there are three models that were floating around this time: the conservative model, the semi-conservative model, and the dispersive model. So at the end of their experiment, which we will get to, they end up discovering that the semi-conservative model is how DNA replication occurs. So just as a note to you guys, conservative and dispersive models, because of this experiment, were proven false. So these theories are not true. The only way that DNA replicates is through the semi-conservative model. So in the next slide, I'm going to draw through what each of these models would look like. And the conservative and dispersive can be a little weird because you're like, how would that even work? That doesn't make sense. Well, it doesn't work that way. The semi-conservative is the one that is true. The others were just possible models and ideas. So whenever I draw it out, don't think that's actually how it's occurring. Those are just ideas. That being said, let's go ahead and draw these out. So I have the conservative, the dispersive, and the semi-conservative models right here. So starting with our conservative model, if this is my old DNA right here, in the conservative model, what it means is that your old DNA always stays together and you have completely new DNA for your other strand of DNA. Whenever DNA replication occurs, we know that the DNA is going to double. So for DNA replication, I'm going to draw two arrows to show that it's doubling. In the conservative model, your old stuff stays together. And the other molecule of DNA that forms is completely new. And then if DNA replication were to occur again for each of these, again, the old would stay together. So on this left one, the yellow is going to stay together again because the template strands stay together, and it just makes totally new DNA. And I'm going to use green to show the new DNA. And in my other one, my blue is now my old. That's going to stay completely together. And for the new one, I'm going to show green again. I'm using green. I'm using different colors because if I were to use blue again, then it would just, you wouldn't really be able to tell that it's the conservative and your blue staying together and stuff like that. So that's your conservative model. Every single time your old DNA stays together and you have completely separate new DNA. In dispersive, we start with our DNA strand and we know we're doubling. And dispersive is a little bit weird. What it basically says is that we chop up all our old DNA, we make new DNA, and then we randomly pull from the old and the new DNA to make our combination of the old and new DNA. So what this would look like is something like this, where, oops, that was the wrong color, blue. So you can see at this point, in our conservative model, we had a strand that was totally old, strand that was totally new, and beneath that, the same idea, and there was never, ever, ever any mixing. In the dispersive model, we do have mixing. Everything is a mix. Both new DNAs that were formed are a mix. And there's going to be a difference in this step with the semi-conservative step in a second. So just keep that thought in the back of your head that this is a mix, and we'll come back to it in a second. So let's go ahead and keep going on our dispersive model and have both of these replicate now. And again, I'm going to use green to show the newest DNA. So now yellow and blue represents old DNA. We're going to chop it up, and then we're going to add some green in there. So I'm going to have some yellow everywhere. I'm going to have some blue everywhere, and the yellow and blue, remember, is representing my old DNA. And then I'm going to add in some green to show my newest DNA. Oops, that needs to be there. This is getting really tight, so sorry about that. Okay, so it's really close together, but you can see there's 4 new DNA molecules, and each has a mix of blue, yellow, and green, where blue and yellow, in this case, was our old, and green was our new. Okay, and the other thing just to mention is that if you look at the proportion of yellow every single time it decreases. So in our top one, it was all yellow, and the next one, it was about half yellow, and the next one, it might be like a fourth or an eighth yellow. So it goes down every single time. So our original DNA decreases over time the amount that we have in each DNA strand. So now let's draw the semi-conservative model. So in the semi-conservative model, again, we're going to start off with that DNA. And since it's DNA replication, we know we're going to be duplicating our DNA. In semi-conservative, you're going to have elements of both the conservative and the dispersive. You're going to have elements of the conservative in the case that each strand is going to be the same type, and elements of the dispersive where the molecule is going to be a mix. In semi-conservative, each of the strands separates and a new strand is built off of that. So these 2 strands are going to separate, and we're going to build a new strand. So now I'm going to compare the 2 stages of, or the second stage of both dispersive and semi-conservative. Notice they're both half yellow and half blue. That's the same in both of them. And they're both a mix. The difference is what that mix looks like. In dispersive, every single strand is a mix. While in semi-conservative, each strand is not a mix, it's the entire molecule that's a mix. So in dispersive, the entire molecule is a mix and every single strand is a mix because every single strand has yellow and blue. In semi-conservative, the entire molecule is a mix, but each strand is its own thing. One is completely old, one is completely new. So now let's show each of these undergoing replication. So that means that our top strand and our bottom strand are going to separate. So in that left example, yellow and blue separate, and then we're going to add on our new green DNA. And the other side, again, our yellow and our blue separate, and we add on that new green DNA. All right, so now that we have that, let's go on to talking a little bit more about what their actual experiment was. So let's talk about the ingredients. The important thing is that they had nitrogen isotopes whenever they did this experiment. They used an N14 or N14 as 1, which is regular nitrogen, and they also used N15, which is a heavier nitrogen, also meaning it's denser. So why did they choose nitrogen? Well, nitrogen is going to be included in every nitrogenous base, so A, T, C, and G, which we know is a really important part of DNA replication. So this is a really easy way to say if we add some nitrogen in there, we know it'll get included in our DNA. And just in case you didn't know what an isotope was, an isotope is whenever you have an element, and there's a lot of different types of it based on the amount of neutrons that they have. So in this case, N14 and N15, they're both still nitrogen. N15 just has 1 extra neutron, meaning it's going to be heavier. And they also used equilibrium gradient density centrifugation, which is just a really, really fancy way of saying that they separated things based on density in a test tube. So, for example, since N15 has a higher density, it's going to be in the bottom of the tube. N14 has a lower density because it's lighter, it's in the top of the tube. So just as a quick example of what this would look like, if we have our test tube, N15 is heavy, it's at the bottom, N14 is light, it's at the top, and if you had a line that was in the middle, exactly in the middle would be half 14 and half 15. And if it was somewhere else in the middle, but it wasn't exactly in the middle, it would show that you have some sort of different proportion of 14 and 15. So for example, if it was located right here, so above the middle mark and closer to 14, that means you still have a mix of 15 and 14, but you have more 14. And if it was closer to 15, that means you still have that mix of 14 and 15, but you have more 15 included. All right. So now let's talk about what they did. So first, they made a bunch of DNA with N15, meaning that every single molecule of DNA had nitrogenous bases in it that had N15. Then they removed the DNA from the N15 and they added it to a new vial that only had N14 in it. And since these DNA is going to be replicating and nitrogen is really important, that means in this new vial of N14, replication is going to be occurring. And since there's only N14, that means any new nucleotides added, meaning any new DNA that's created, has to contain N14. And any old DNA from our previous vial would have N15. Okay, so now let's talk about the results. So I'm going to click through that really quick. And I'm actually going to draw it out and leave this for you guys to come back to and look at if you need to. This is basically just explaining what it looked like in the tubes. We're going to talk about that and then show that it is semi-conservative. So these are the results, which is explained in writing on the previous slide. And at the end of their experiment, they discovered that it's semi-conservative. So knowing that, I'm going to draw what it would look like. So hopefully it makes a little more sense why it looks like this in the tubes. So to begin with, we started out with some N15 DNA. And then we added it to N14. So we know in semi-conservative, our strands are going to split. And we're going to add on N14 DNA, which I'm going to represent with orange. All right. So now in this case, every single DNA molecule has 115 and 114. 14 is light, 15 is heavy. So if we have 114, 115, they'll meet somewhere in the middle. So that's why you see that pink line where it's not at the top, it's not at the bottom, it's somewhere in the middle to show that that was a mix. So at this stage in the experiment, they were able to rule out the conservative model. Because remember, in the conservative model, there's never any mixing. It's old, it's new, there's never any in between. So because we had an in between, they knew at this point, okay, it's either dispersive or it's semi-conservative. And it's the next one that told them for sure it's semi-conservative. For dispersive, you would, because, let me go back to the pictures. Notice that in the dispersive one, every single DNA molecule looks about the same, meaning that there's never going to be two lines. Every single DNA molecule is going to be at the same density. So in our next vial, it has two lines, which is why they didn't know they knew it wasn't dispersive, because with dispersive, you'd only have one line because every single one is the same density. All right. So now to show that this is semi-conservative. Remember, in semi-conservative, your strands are going to split. So for this top one right here, that green would go up, the yellow would go, the orange would go down. And then we're going to make new DNA. In this case, because it's N14 DNA, which I represent with orange, I'm going to add another orange line to both of these. And then I'm going to do the same thing on this bottom one. Split up those two strands. So there's my green one, here's my orange one, and then I'm going to add new DNA to it. So what you should notice here is that we have two that are that mix in the middle, and now we have two that are completely new DNA. There's no more mix anymore. It's all N14. That's why it's at the top, and that's that orange line at the top. So there's something weird in the next one now, and that orange line got really, really fat. And that fatness doesn't mean anything about heaviness. All it means is that there's more of it. So it's talking about concentration or amount. A fatter line means there's more N14. than the mix. So let's demonstrate that. I know that whenever I am replicating, each of these is going to split up. So I would have green go to one spot, yellow go to the other, or sorry, orange go to the other. And then I would add an orange to each of these. In this next one, those two oranges would split up, and then I would add an orange to each of those. In the next one, that green and the orange would split up. And then I would add an orange to each of those. And in this last one, those two oranges would split up, and then I would add an orange to each of those. So now let's look at the amounts of these. For my ones that are a mix, you'll notice there's still only two. We started with two that were a mix, and then we had two that were a mix, and now we have two that are a mix. So every single time we replicate from vial 2 all the way on, we always only have two of these. So that's why that line stays the same thickness, because there's only two. In contrast, looking at your ones that are totally N14, you had two at this stage, and now you have 6. So you can see there's a large increase in the amount that we have, which is why that is a thicker line now. And so because of these results, they discovered that DNA was replicated in a semi-conservative manner. All right, so in the next video, we'll talk about leading and lagging strands.
I hope you found this video really helpful. The concepts and information presented in these videos will be true no matter what genetics class you are taking. However, the concepts presented in this video are referencing material currently covered in Baylor University's coursework. Remember, if you are currently enrolled as a Baylor student, we offer free tutoring services. Our tutoring center is located on the first floor of the Sid Richardson building. You will find all the details you need to know about these services on our website, www.baylor.edu/tutoring. You may schedule a free 30-minute one-on-one tutoring session online through Navigate, or just drop in during our open business hours. For more information about our current services, please visit our website. Thank you.